today is set some new data in the context of old information we had, and how we intend to go forward looking at calcium signaling in exocrine glands. So this one is not necessary. So just for people who don't know, I'm in the University of Rochester. It's a smallish, research-intensive medical school in upstate New York. We were founded by George Eastman, who also was the founder of the Kodak Film Company. And let's hope we don't go the way of the Kodak Film Company. Rochester is in New York State. It's on Lake Ontario, essentially about 300 miles from New York City and about 90 miles from Niagara Falls. So that geographically puts us kind of in the middle of New York State, between Buffalo and the city. So my lab for many years has worked on exocrine glands and exocrine secretion. And this is a sort of low magnification micrograph of what exocrine glands look like. I always think of them as looking like a bunch of grapes, where there are two cell types: the acini, which are the secretory cell type, which secrete things into a duct, and here the stalks of the bunch of grapes are the ducts. So at a higher magnification, this is a immunofluorescence showing the acina structures emptying into these ductal structures. What we're saying here in green is TMEM16A, which is a calcium-dependent chloride channel, which is responsible for the fluid secretion from many exocrine glands, including pancreatic acina cells and salivary gland acina cells. So, why do we work on these glands? We work on these glands essentially because everything these glands do is dependent on an increase in cytosolic calcium. So when calcium goes up, there are effectors which are important for secretion of proteins through exocytosis and for secretion of fluid. So over the years, we've concentrated on working on salivary glands and pancreatic exocrine pancreas and pancreatic acina cells, and tried to compare and contrast the calcium signals that we see in these glands. So in terms of fluid secretion, the calcium-dependent effector is this chloride channel, TMEM16A, as well as this potassium channel, which is IK or BK. And the way that you actually generate saliva or fluid from any of these exocrine glands is you have to move essentially chloride from the basolateral space into the lumen. So there's essentially a transepithelial flux of chloride. And this is accomplished by chloride being dragged down up its electrochemical gradient with a transporter called the sodium potassium to chloride cotransporter that drags chloride into the cell where it accumulates. This is all powered by the sodium potassium ATPase. And then on stimulation, nervous stimulation, and in terms of salivary gland, this is the sight and smell and taste of food, neural regulation results in an increase in calcium, which activates the chloride channel, increasing the flux of chloride into the lumen, making the lumen negative. Sodium follows paracellularly, and we have a sodium chloride-rich solution. And then water follows osmotically through the tight junctions and also through the cells. The calcium-dependent effector, predominant calcium-dependent effector, is the chloride channel. But to maintain the membrane potential away from ECL and maintain chloride flux, essentially the membrane has to be repolarized, and it's repolarized by calcium activating a potassium channel, calcium activated potassium channel. So we can see the beautiful polarization of, hopefully we can see the polarization of the localization of the chloride channel, which is in green, and the sodium potassium ATPase, which is actually in red here. I'm trying to get this movie to play. It possibly will not play because it's a pointer, not a pointer. Yeah, there we go. So here's the three-dimensional reconstruction of the potassium channel, of the sodium potassium ATPase in the chloride channel. You can see the beautiful polarization. So the signal transduction pathway upstream of the calcium signal is the canonical G protein coupled receptor pathway through GQ. This is a nice slide from one of Martin's reviews, basically showing the canonical pathway. And in the case of salivary glands and also pancreas, acetylcholine binds to its 7-transmembrane receptor, activates GQ, phospholipase C beta, and produces IP3, which binds to its receptor and results in an explosive release of calcium. So IP3 receptors are incredibly important for all these processes, and we know that from work from Nikoshiba, where he basically knocked out the type 2 and the type 3 IP3 receptor, which are the predominant IP3 receptors in salivary glands and in pancreas, and showed that these animals were born normally but died soon after weaning, and they died soon after weaning because they had no calcium signals in their salivary gland acinar cells, so were not secreting any fluid and therefore couldn't assimilate dry mash food. Even when you fed them wet mash food, the animals failed to thrive, basically because their exocrine pancreas is not working to secrete digestive enzymes. So the IP3 receptor is incredibly important. So over the last 10 or 15 years or so, many groups, including my own, have accumulated data relating to the characteristics of the calcium signals that you see on stimulation of these cells. And the vast majority of this work has been done conveniently in isolated acini, enzymatically digested acini, or in slices or lobules from the pancreas or salivary gland, but they've all mostly been done as ex vivo preparations. And so we know a lot about the calcium signals. When we stimulate either salivary glands or pancreatic acini with acetylcholine analogs, we see beautiful calcium oscillations, which increase moderately in their frequency with concentration. And these oscillations are fairly slow, 3 or 4 oscillations, possibly a minute. So there's temporal information that we've gathered from doing experiments on isolated cells and small clusters of acini. We've also gathered a lot of spatial information. So this is again just imaging relatively slowly, but stimulating the cells with acetylcholine. And what you can see is that obviously the signal is not homogeneous. There are calcium signals that appear to spread between cells, but typically the signals are actually global and relatively slow. These are the characteristics that we've kind of established working on isolated cells. So there's also a lot of spatial information that's encoded in the signal transduction pathway, and that's the exemplar is here. Essentially, what we're doing in this experiment is uncaging IP3 locally in the basolateral end of a cell, essentially under this red dot, and then monitoring the calcium signal after activating the laser. And when we do that, you see that's probably mostly a laser artifact, but the calcium signal essentially initiated in the apical end of the cell. So we'll play that again. So again, uncaging in the basal end of the cell, there's basically a gap where nothing much happens, and then the signal is initiated in the apical end of the cell and spreads in a wave. Now, the most important thing that we have shown in salivary gland acinar cells is that the signal is invariably global. So whenever we initiate a signal, initiates in the apical end of the cell, but rapidly globalizes, and that's shown here. So uncaging different amounts of IP3 and monitoring the calcium signal in either the apical or the basal regions of the cell. The signal initiates in the apical end and spreads in a wave towards the basal end of the cell. And so that's a characteristic of signaling that we have laid our hat on in salivary glands, and we rationalized the spatial aspects of the signal as being necessary for activating spatially distinct effectors, the chloride channel on the apical membrane and the potassium channel on the basolateral membrane. And just make a quick point here that this was actually different in pancreatic acinar cells. I don't have time to show you, but in pancreatic acinar cells, as people have shown, many people have shown, in particular the Peterson-Topekine group, that calcium signals can be local in pancreas and don't spread. So there was a difference that we thought that we could see in salivary glands and pancreas, which related to the function. So if we look at the IP3 receptor in salivary gland acinar cells or pancreatic acinar cells, there is an intimate relationship between the IP3 release channel and the chloride channel, which is the effector. So this is a super resolution image, essentially showing you the localization of IP3 receptors in the ER membrane, close to the apical plasma membrane, and the localization of the chloride channel. And actually, these data show us that the two channels are probably about 50 to 75 nanometers apart. There's an intimate structural relationship, physical relationship. There's also an intimate functional relationship, in that now when we globally uncage, we see that the chloride channel is activated, essentially follows faithfully the calcium signal in the apical end of the cell. Okay. All these experiments, as I said, are done in isolated acini or single cells or lobules. And the issue with that is that obviously the signal is, we're measuring the signal, but not in its native environment. And indeed, it's even compounded by the fact that the way that we apply the agonist to the cell, which is basically a square wave pulse of acetylcholine or carbachol, is not what the cells are going to see when they are stimulated by nervous stimulation. So we were motivated by some experiments we were doing to try and see what happened to calcium signals in disease states of salivary glands where secretion seems to decrease, to have a look at these signals in vivo. So we've set up a platform to do intravital microscopy in salivary glands. So the way we do this is animals are subjected to some surgery on an inverted, and then put on an inverted microscope. The submandibular gland is exposed. There is some saline around the submandibular gland. Saline is the immersion material for a 20x objective. So here's the mouse. Submandibular gland is isolated. Here's the mouse on a warming plate. We keep them alive for 2 or 3 hours. And just to show you how deep we can get into the cells, this is a mouse that expresses constitutively membrane-targeted tomato. It has a stop codon, which will allow you to express, in particular cells, a GFP when the stop codon is excised by an appropriate Cree. And essentially, here's the Z-stack through these membrane tomato-labeled cells. And essentially what you're seeing here are mostly acinar cells. And then as we get deeper into the tissue, you can see ducts appearing. So basically every cell in the body in this mouse is labeled with a membrane-targeted tomato. So our approach is to measure calcium in these animals has been to cross an animal which expresses a floxed, a stop codon around a GCAMP, a relatively fast GCAMP, GCAMP6F, with a promoter driving a Cree that will express the probe in particular cells. So we've done this For acinar cells with a MIST promoter, for ductal cells with a K5 promoter, for neural cells with a acetylcholine transferase promoter. I'm just going to show you the acinar cell data. So we cross heterozygote mice, we get MIST GCAMP compound heterozygotes. It's inducible. We give the animals a tamoxifen gavage, and a week later we can express GCAMP6F, in this case in the acinar cells. You can see that it's only in the acinar cells. The ducts are now big black holes. So first things we did were optimize the system on a relatively old multiphoton microscope, where essentially what we're trying to do is we've wrapped gold electrodes around a blood vessel and nerve bundle that goes into the submandibular gland, which we were hoping would isolate the submandibular nerve, and then we stimulate the submandibular nerve. And so here what we're doing is it's 30 hertz for a second. The excursion is 200 microseconds at 50 milliamps. So here we go. Basically, it's relatively clear when the signals were responding. So we can actually stimulate this nerve. That's good news. The thing that was troubling us at this time was that the resolution is not particularly great, but we can increase the frequency, and we can increase the frequency of the stimulation and increase the amplitude of the signal, as you might expect. As we increase the amplitude, we increase the frequency of stimulation, the amplitude increases. If we monitor the signal so mice have two submandibular glands. If we monitor the signal in the contralateral gland when we're stimulating the ipsilateral gland, there's essentially no signal or very little signal, which indicates it's not just passive charge transfer through the saline that's bathing the tissue. We can increase the length of time that the animals are stimulated with a particular frequency. This is 30 hertz for various times, and you can see the signal gets longer, as you might expect. There's presumably more neurotransmitter released, and we can take the animals and actually sort of keep stimulating them. We can pace the gland just by repetitive stimulation. So yeah, and we can do this for quite a period of time. So this has allowed us to set up the method. What it didn't allow us to do was really get any particular information from the in vivo situation, because the resolution wasn't so good. So we turned to using a microscope in our imaging core, which was a new multiphoton microscope equipped with a resonance scanner. And now we can actually get really quite good subcellular localization. That's 40 microns at the bottom here. And we can actually investigate the impact of increased frequency of stimulation on the calcium signals quite easily. So now we're stimulating this in this experiment with 1 hertz for 12 seconds with this excursion. And you can see now we're recording at 10 frames a second and averaging 3 frames. So it's 30 frames a second, but averaging 3 frames. And you can see that we now start to see some calcium signals in a minority of cells at this low frequency stimulation. We can manually put regions of interest onto cells in an image J, get the readout of the calcium signal. If we increase the frequency of stimulation, this is in the same field, you'll see that now we're recruiting more cells to respond. And again, the cells are the field is same field is stimulating for 12 seconds, and you can see we start to recruit some spikes. Notice that these are much faster than we saw in the isolated tissue. And if we go up even further, this is 5 hertz stimulation, same field. More of the field of view is being recruited. And actually the amplitude of the signals is larger. See here again, we're seeing some spikes. And finally, if in this series, if we go to 10 hertz, basically the vast majority of cells in the field will respond. Yeah. So this was sort of a challenge for us to actually do some analysis of what we were seeing in the fields of view. And I could not persuade a postdoc to put 100 and whatever regions of interest over all the cells in the field of view. So we decided we'd come up with a sort of a batch analysis way of analyzing these images. And what we decided to do, at least initially, was to divide the field of view into a grid. And we settled on an 8x8 grid, which is basically giving you 32 microns dimension for the grid, and then average the signal within the cells in the grid. Now, I mean, obviously we're not capturing all cells, but it's giving us an idea of spatial differences in the cell and capturing a lot of signals. So then we process the image, essentially produce a standard deviation image or a maximum intensity image, and then use that to read out the information in the 64 grids. And so here's the different stimulations. So essentially what you can see is that at 1 hertz, there was a few cells that or areas that responded here, more in the 3, 5, and 10 hertz boxes. And obviously the amplitude is getting bigger as we increase the frequency. And you can see the readout from the 64 boxes in the bottom of this slide. So essentially what we seem to be seeing is an amplitude modulated system. As we stimulate for longer, we get bigger signals. And just to sort of validate what we do with the batch analysis, if you just simply pick, these are random regions of interest that were picked manually versus the sort of non-biased measurement, you've obviously lost a lot of the temporal information, but it seems to faithfully reflect the amplitude increase in the signal. So if we analyze a whole field of view, what you basically can see is if you bin the amplitudes as you increase the frequency, obviously more of the grids are falling into the higher changes in signal, and obviously at lower stimulus, even a majority of the cells are really not responding very well, and the average in those grids is basically amplitude dependent. The other thing that we see is that there's a recruitment of responsiveness to cells, to individual grids, in that the latency of response seems to decrease markedly. So at 1 hertz, the cells that do response have a long latency, which before they actually fire, and as you might expect, as you increase the frequency, the latency becomes very short. If you analyze a bunch of mice, you can see that that's basically borne out. The mean peak height increases with stimulus. The number of grids that respond increases with stimulus frequency, such that between 1 and 3 hertz, you essentially recruit all the areas of the field of view. I should say that, you know, an obvious question is whether these frequencies correspond to how the nerve is firing in vivo when the animal is wandering around sniffing cheese. And all I can say is that between 3 and 10 hertz, this animal is salivating on the microscope stage. So it seems to be an appropriate stimulus. And again, here over a bunch of mice, the latency will decrease as you increase the stimulus threshold. So as you notice, there was some heterogeneity in where cells respond on the grid. So in the example I showed, this area seems to be more sensitive, but we see that in multiple image views in multiple mice, that particular areas seem to be much more sensitive to the low frequency stimulation. So what we did to investigate that was essentially make a max projection image and plot the grids that respond greatest and rank those for amplitude. So essentially, based on the 5 Hertz stimulation where everything is responded, the grid that responds the greatest is G6 in this case, and this is the grids ranked by increasing maximum amplitude. And now in this one field of view, if we go back and look at the 1 Hertz, 3 Hertz, and 10 Hertz stimulation, they essentially follow the same rank order of responding grid. So it appears that in this case, this area was more sensitive to stimulation than the rest of the field of view. And that's held up in multiple mice. So there could be several explanations for this. I mean, one explanation could be that there is not a homogeneous distribution of nervous input into the gland. Another possibility is that there is a homogeneous, basically, distribution of nerves, but neurons are recruited to fire as we increase the frequency. Or it could be that some acini are actually more sensitive to homogeneous response of neurons. So we're starting to investigate that. I mean, we've crossed our cholinesterase mouse with our GCAMP6F mouse to give us a calcium indicator in parasympathetic neurons, and that data is coming online. We've also crossed this cholinesterase transferase mouse with the MTMG mouse, which again constitutively expresses membrane tomato or tomato in each cell type, and when the stop codon is excised by an appropriate CRE, will express GFP. And so this animal now expresses GFP in its parasympathetic neurons, and basically you can see that there's a fairly homogeneous distribution of parasympathetic neurons. So we're currently investigating the other 2 options that I mentioned. So one thing that you may have noticed when I showed you these images are that many of the signals, especially at low frequency, are actually quite local, which is very different to data that we obtained in isolated tissue. So much of the signal, and these are standard deviation signals, appear to originate, but also are sustained, maintained around the apical membranes of the cells. And here's a higher magnification movie taken at 20 Hertz. And basically you can see, especially in this acini here, which seems to be in a flat plane around the lumen, that these signals are very local. And I think this is 3 Hertz stimulation. And the signals essentially never globalize. So you can make a standard deviation image that would focus on this acini, essentially here you can see this volcano plot essentially showing you that the signal is much greater in the apical end of the cell. You can obviously manually put on regions of interest, and you can see that essentially we're getting these oscillations in the apical end of the cell and essentially nothing in the basolateral regions of the cell. So this is distinctly different from what we saw in isolated cells. So we had to come up with a way of analyzing these data, and this was essentially done in collaboration with my long-term collaborators in New Zealand, James Sneed and John Rugas. And essentially they created a MATLAB script that would allow us an average image to make an apical mask, take the apical mask, generate the signal in in each of these samples in the apical mask. And what this is doing is plotting the change in every pixel within those regions of interest, and then the black giving you the average. From the average, you can take these signals and output the response in the same area, in the same region of interest to the various stimulations. And so you can, this is a very, this is a very labor-intensive way, labor-unintensive way of analyzing these data, and really has facilitated us being able to get some meaningful information out of these traces. So again, here you can get the average of the averages here. So when we've done this, at least at low frequency, lower frequencies, we're getting a modest increase in the increase in the oscillation frequency. And I think this goes with the fact that this seems to be more of an amplitude-based system that's stimulating the secretion. And so we're clearly not pacing the calcium response with the input. The frequency of the calcium oscillations is obviously much slower than we're pacing in the majority of cases. And I think sort of our working model for what's happening is that when we stimulate the nerve at low frequencies, essentially the neurotransmitter builds up and decays as it's metabolized, and at a point it'll get to a threshold where it can actually fire a signal, and then the inherent oscillator in the system is essentially activated. And as you increase the frequency, essentially you're not allowing the neurotransmitter to decay as fast, and the latency shortens, and again you stimulate the increase in signals. Last couple of slides. So the neurotransmitter, as you might expect, is likely to be acetylcholine, and evidence for that is that we can inject pilocarpine, which is a partial agonist of the muscarinic receptor, IP into these mice, and we can evoke signals which have kind of similar characteristics to the nervous stimulated signal, the neural stimulated signals. And that's basically shown here. Obviously, we're coming into it without a baseline, but we get spikes. There's apical signals and no basal signals. Further evidence that this is an acetylcholine-mediated response. We can inject the animal with a cholinesterase inhibitor, physostigmine, and stimulate, and you can see that the signals are actually bigger and occur in more cells when the animal has been treated with a cholinesterase inhibitor, allowing acetylcholine to stay around longer. And so this is essentially from that field of view stimulated over different frequencies, and you can see that at least at the lower frequencies, physostigmine markedly potentiates the signal. So in the last 2 minutes, so how does the secretory process function if the calcium signal is localized and is not able to efficiently activate the potassium channel that's on the basolateral membrane? Some insight from this, as I think we've gained from modeling, and this is again with my long-term collaborator James Sneed, and he essentially modeled the salivary acinar cell as an equivalent electrical circuit based on the fact that the basolateral and the apical membrane were separated by tight junctions, which gave the membrane resistance and allowed these membrane potentials to be independent. And what he actually did in his modeling, much easier to do this in modeling than experimentally, is ran simulations where he put some of the potassium conductance in the apical membrane. And what he showed was that between about 10% and 40% of the potassium channel being in the apical membrane would be predicted to increase the salivary flow. 4 or 5 years ago, we tested this idea by locally uncaging calcium in the apical end of the cell. And so Here's the response here. Only calcium in the apical end of the cell, and we could show that that would activate a potassium conductance as well as a chloride conductance, whereas a basolateral uncaging of calcium would only activate a potassium conductance. So there's clearly some potassium channel in the apical membrane, and this would allow efficient repolarization of the apical membrane below the tight junctions to maintain the membrane potential away from the equilibrium potential for chloride and keep the chloride flowing. So that's essentially how we rationalize how this local calcium signal, predominantly local calcium signal, is able to efficiently stimulate fluid secretion. So that's me done. These are the people in the lab, and the majority of the in vivo work was done by Takahiro Takano, who hopefully is on the call and can answer any technical questions that I flub on. And some graduate students in the lab are also working on this in vivo imaging approach to answer some other questions. My collaborators in New Zealand are James Sneed and John Rugas, and a collaborator at University of Rochester is Catherine Overt, who has got me into the mouse farming business. So I'm willing to answer questions. Thank you for your attention.
Thank you, Dr. Yule, for this interesting talk. We had a few questions already. First one from Martin. He wants to know if you load the calcium indicator into isolated cells, would this be responsible for the globalization that you would see there?
Yeah, no, that's a distinct possibility. I mean, we know from numerous studies that the calcium indicator is going to affect the signal. That's a definite possibility. It's not my favorite option. My favorite option is that the gland is losing polarity when it's taken out of the animal, and it's not in contact with extracellular matrix, basal membranes. But I think what we plan. I think what might answer that question is, and we haven't had enough animals to do that, but we will, is to actually prepare acini from these animals and monitor the calcium signals from the GCamp in isolated acini. I think that might be one way to at least get at that.
Indeed. Yeah, it might be necessary because the GCamp might be not sensitive enough to pick everything up, or? Well,
I mean, GCamp, my understanding is KD is about 200 nanomolar, which is kind of similar to Fura, but Fura is doing all sorts of other things, or the AM. Yeah, so yeah.
Okay, next question then from Antonio Villalobo. Is there evidence for intracellular calcium mobilization via gap junction channels?
There is definite evidence in acini cells that calcium calcium and/or IP3 will actually traverse gap junctions and affect the signals in neighboring cells. Mm-hmm. And so in isolated acini cells, we have published some data that shows that if you stimulate a cell, there is one cell that's always the most responsive, and will respond first, and then other cells will respond in a coordinated pattern. And we've always thought that was dependent on gap junctions, and it sort of goes along with, I think, work from Jan in the past, which showed that with single cells there was basically, as I think it was, as groups got bigger, you recruited more responses. I think, yeah, I'm trying to pull that out of somewhere where it doesn't belong. But, yeah.
Okay. Another question from Tofik Rahman. Is it known whether ATP can also be co-released with acetylcholine in this system?
I think that's highly likely. I think it's highly likely that that you stimulate these neurons, and you're stimulating either neurons that are releasing acetylcholine and ATP co-packaged, or neurons that are releasing ATP on their own. And so, one way of addressing that is we've thought about doing sort of similar experiments as we did with the Pfizer stigmine, and that's would be bathing the tissue, not injecting the animal, bathing the tissue with a pyrase, and seeing what that does to the signal. I mean, that's really interesting because, I mean, we've done parallel experiments working on duct cells where the probe is expressed in duct cells, and the signals are completely different, and they're slow, and they look like they're paracrine signals, and duct cells are known to respond to ATP very robustly, and that's my bet as far as the duct cells go.
Okay. Sandy Patel wants to know if there would be an effect of the anesthetic that is used.
Yeah.
On the calcium signals.
Yeah. I mean, the anesthetic is ketamine xylazine, and that's again a definite possibility. I mean, I don't know about ketamine, but most anesthetics affect sodium channels and excitability. Clearly, we can still excite the neurons, but I think that, again, is something that we have to think of. Yeah, it's a tough one. Yeah.
And Anthony Morgan has a question on whether or not there is a mitochondrial belt in these cells as compared to in the pancreas, and would this help to restrict the apical response,
So we, I don't know whether I don't think I've got the slides here, but we have been doing experiments imaging mitochondrial membrane potential, NAD, NADH in this same preparation. And in salivary glands, there is no mitochondrial belt. We have some information because obviously the MIST, the MIST promoter will express this indicator in pancreas as well. So it'd be interesting to see if there are any differences in the calcium signals there than what we've all documented in the past. But I mean, even in isolated cells, we published maybe 10 years ago with Jason Bruce that any mitochondrial belt that was seen in isolated acinar cells was not in salivary glands. And again, that was another thing that we put down to facilitating these global signals. That's a good question. The mitochondria, at least in isolate, so this is what worries me, that the mitochondria in isolated acini seem to be wrapped around the nucleus more than anything. When we look at mitochondria in tissue in vivo, they're actually just essentially lining the apical and lateral membranes, plasma membranes. So clearly the localization is different in tissue versus in isolated cells. So we're sort of learning some lessons.
Yeah. Then Martin Bootman was wondering if it is an amplitude modulated secretory system, or is it a summation of cells that are individually frequency modulated?
Yeah, and that's a very good question. I mean, we our conclusions there come from modeling more than anything. And we've published a number of models which have been counter to my what I would have preferred, and that is that calcium oscillations, even though they're beautiful in these cells, don't it doesn't matter what the frequency are for secretion. And at least in individual cells, the data that we had from individual cells, the model didn't care whether the cells were oscillating or not. It was simply the amplitude of the signal for the time that it was elevated. And I mean, since we've had these data that show local signals and faster signals, our model of secretion, which we developed with James, has been modified. But it still relies on the fact that it's the amplitude of the signal rather than the frequency. I think an adaptation of what Jan was saying is it maybe could be recruitment of cells and their amplitude to give an integrated calcium signal over more cells.
Another question from Anthony Gaglione. Can you measure amylase release in the in vivo system, or do you need larger calcium signals for amylase exocytosis?
Yeah, That's a great question. I mean, we potentially can. I mean, we're gearing up to actually measure fluid secretion from these animals that are on the stage of the microscope. This is something that we'd like to develop. The animals are salivating when we stimulate them. We just have to collect it. If we can collect the saliva, then we have pretty sensitive ways of measuring the amylase in the saliva. So that could be done. But remember, Anthony, that although amylase secretion is stimulated by acetylcholine in salivary glands, the signal that stimulates exocytosis in salivary glands is actually cyclic AMP. But we would be stimulating sympathetic nerves into the gland as well, in likelihood.
Indu Ambudkar asks if you have any comment about where the muscarinic receptors are localized. And the neural network is pretty extensive all over the acinar cell of the gland. Yeah. So where are they?
Yeah. Well, I mean, again, information from isolated cells and small groups of acini would say that they are above tight junctions, but possibly close to tight junctions. But I'm not sure what that data actually means, and if it's not done in isolated in glands. Now, Indu probably knows. I don't recall people staining for muscarinic receptors actually in tissue slices. I think I've only seen it from Schmull in isolated acini. Preliminary data from imaging GCAMP expressed in axons going to these acini suggests that calcium signals are, where acetylcholine is released is very localized.
Okay. Then Joseph Heiser wants to know if there are differences in IP3 receptor or potassium channel types between mice and human. And can you grow human salivary gland organoids ex vivo from stem cells to compare the calcium signaling?
I think work that Indu and I did suggested that the IP3 receptors are the same in humans versus rodents. So it's predominantly type 2 and type 3 receptor in humans and rodents. A potassium channel, I am not so clear on. I mean, mice have IK and BK. So they have a large conductance calcium maxi-K channel and an intermediate conductance potassium channel. To sort of address the last part of the question, organoids have been grown from salivary gland tissue, and they sort of recapitulate, and calcium signals have been measured in these organoids. The polarity of them is not quite as defined, or really not as defined as even to isolated acini clusters that you make. So I think you could measure calcium from them, and there are people trying to do it. There are people, Catherine Ovett in my institution is trying to generate a salivary gland on a chip, and you can actually measure calcium signals in those cells that they culture as organoids.
Okay, and maybe a last few questions from Geert. He wants to know if the heterogeneity of the cellular responsiveness is related to the heterogeneity of IP3 receptor type 2 or 3 expression among different acinar cells.
That's a good question. That's a possibility. I mean, when we've stained, again, we've stained tissue slices, the type 2 and the type 3 IP3 receptor, they look like they are pretty homogeneous in acina slabs. It's tough to tell though, because you think about this bunch of grapes, and all you're doing is taking a slice through the bunch of grapes, and so there are some cells that don't look like they have IP3 receptors in them because you haven't captured the apical end of the cell. So that's tough to answer definitively. I'm thinking the heterogeneity is much more likely to be due to heterogeneity of acetylcholine receptors are much more likely, as my chair keeps telling me, Bob Dirksen, that I should learn lessons from muscle. And that essentially what we're doing with increased frequency is recruiting more nerves to fire. And therefore spreading out, the signal now becomes more extensive because there's more acetylcholine spread out across the whole field.
Okay. Yeah, thank you for your time and for answering the questions. I think this more or less concludes the official part of this talk. Dr. Yule has agreed to stay on for a few more minutes, if I recall, to answer a few more questions after the talk. So I would like to thank, obviously, the organizers for hosting these sessions.